Now, let's continue to study the nucleic acid and DNA and RNA polymers and how DNA and RNAs are different, and the building block of this is a polymers of nucleotide. So before that, we want to study first biomolecular basic concepts of interaction. So here I put the two examples of water molecules and H2O and H2O. As these, as I said, water, hydrogen has a partially positive and oxygen is partially negative. So one water molecule is partially negative can attract by the another molecule of this proton or hydrogen, which is partially positive. So these bonding we call as a hydrogen bonding, typically because there involves hydrogen, and that partially positive hydrogen atom in a polar molecule is attracted to a slightly negative charged negative atom in a neighboring molecule. And a good example is a water molecule. So this is a simple diagram, and this is more like three-dimensional interaction. You can see this one hydrogen, this water molecule, and the other neighboring water molecules interaction. So water is a fascinating molecule that it's so small, tiny, however, it has very high boiling temperature. That's because of this hydrogen bonding. And hydrogen bonding is only 1 to 5 kilocalorie per mole compared to covalent bonding, which is a shared electron between this example is oxygen and hydrogen. And this covalent bonding is actually much more energy. For example, like 50 to, let's say, about 300 kilocalorie per mole. So it's, it's orders of magnitude bigger. However, there's so many hydrogen bondings in water that makes the water high boiling temperature, and water has a very high surface tension. So this gives, and the waters compose over 70% of our body. So it's very important that hydrogen bonding is important, as you already seen the protein secondary structure, and that alpha helix and beta pleated sheets are due to the hydrogen bonding. So now let's discuss about the building blocks of this nucleic acid or DNA and RNA that we call as a nucleotide. So nucleotide in a simple form is this diagram. Very simple. You see there's a pentose with 5 edges. You remember Pentagon as a representative building in the USA, and that shape is this, 5 angles. And so we call it as a pentose, and tose is a sugar molecule. And there's one phosphate group, and there's another, it's an organic base. So nucleotide is composed of a pentose. This is a 5 carbon, this is actually carbon, 1, 2, 3, 4, 5 carbon sugar molecule, and one phosphate group represented as a P, and the other is an organic base. So this organic base actually makes each subunit of DNA and RNA. And how does DNA and RNA different? From its structural aspect, this pentose, the sugar molecule types are different, and that is reflected in their original name. And DNA stands for deoxyribonucleic acid. And that contains, of course, deoxyribose sugar. Then what about RNA? RNA is ribonucleic acid, and it contains ribose sugar instead of deoxyribose sugar. We'll discuss about the fine molecular difference later on. So these building blocks of DNA and RNA, and that DNA and RNA we call nucleic acids. And nucleic acids are polymers of these nucleotides. And nucleotides are so monomer for nucleic acid polymers, and there are a number of kinds of nucleotides. Very simple, there's 4 different types. One is called adenine, guanine, cytosine, and thymine. It's for DNA. And RNA is only a slight difference of this thymine becomes uracil. So we call ATCG or AUCG. And the importance of this nucleic acid is it gives the solution of how genetic information can pass down in many of the life forms. So it provides a mechanism for the storage of genetic information within our cells. And there's a phosphate group, and there's a pentose 5-carbon sugar molecule, and organic base. And this organic unit is composing nitrogenous bases, and that's A, C, T, and uracil. So the biological macromolecule of with using amino acid, we form a peptide, and this peptide chain will compose of a protein structure, and that diversity comes from amino acid, and these monomers has about 20 naturally occurring standard amino acid will generate a multitude of these peptide chains. And what about DNA and RNA? As I said, DNA and RNAs composed of this nucleotide. And nucleotide has this 1, 2, 3, 4 subunits. And that will compose of our DNA and RNA diversity and its information content. So as we see previously, this phosphate, I want to just discuss a little bit of this phosphate ions in biochemistry. So this PO4 3- is very important in biochemistry. So phosphate is an inorganic chemical and a salt of phosphoric acid, which is H3PO4, and which can be decomposed as H plus H2PO4 minus, and this becomes H plus HPO4 2 minus, and this will become also H plus PO4 3 minus. And this phosphate ion in biological system is a free phosphate ion. It's called, we call inorganic phosphate, and we denote it as a P sub I. At physiological or neutral pH, consists of a mixture of, as you see this in chemical reactions, HPO4 2 minus or H2PO4 1 minus as primary consists of physiological pH. And these phosphates are commonly found in the form of, you may heard a lot about like ATP, right? So adenosine phosphates. And so such as AMP, ADP, ATP, and in DNA and RNA, phosphates are commonly found. And why is phosphate so important? Because that bonding to make this contains a very high energy. So it can be released by hydrolysis of ATP or ADP, and that can create high amount of energy. So you probably heard the ATP, adenosine triphosphate, is a currency of our life. So the addition and removal of phosphate from proteins in all cells is the main strategy in the regulation of metabolic processes. So when you are studying cell biology, especially molecular cell biology, you will realize a lot of this cell signaling is involved in this phosphate. And typically in the laboratory, when you are dealing with the cells and tissue, and once, for example, you take out brain tissue from a sample, then usually put this one not in water, because that water has a different pH and concentration. So we typically use this phosphate buffered saline. So this buffer serves as keeping the pH in the neutral form, usually in biology, body, like 7.4. So this PBS is commonly used buffer solution in biological research to help maintain constant pH. So to preserve the physiology of the cells or tissues of the sample. The osmolarity and ion concentration of solutions match those with human body, and we call this isotonic. So next time we will try to understand more of the central dogma of molecular biology. Okay, thank you for your attention.